typical, actually we're not talking about any cocci, so all gram-positive rods. Bacillus and Clostridium are the ones that will take the majority of our time in this deck. And we'll only talk about Bacillus today. We'll talk about Clostridium starting tomorrow. Corynebacterium will not take a lot of time. We'll just talk about diphtheria. Corynebacterium diphtheriae is the most significant organism here. And Cutibacterium used to be a Propionibacterium, causative agent of acne. Not a big deal, but something you'll probably want to be familiar with just because you'll likely see a lot of it. Actinomyces, Actinomycetes, that causes some significant pathology. So we'll spend a couple slides talking about that. And then Listeria. Listeria is one that we talked about briefly as causing meningitis. So that is another significant pathogen and associated with some big outbreaks there. Also in this deck, at the very end, we'll actually talk about some good guys for once, because it's good to know as clinicians, particularly when you have dysbiosis in the gut or in some other areas that are colonized and require good bacteria to be there. Lactobacillus and Bifidobacterium, two very good bacteria, occasionally implicated in disease, very rarely though. Most of the time, these are the ones that you want to make sure those populations persist and don't get wiped out through broad-spectrum antibiotics and things like that. Okay, we talked about spores a little bit in block 1. We talked about spores again a little bit in block 2 when we were talking about how hard it is to disinfect spore formers. But the good thing is most bacteria cannot form spores. Only these two genera of gram-positives can. So Bacillus, which is an aerobic genus, and Clostridium, or Clostridioides as it's been reclassified, which is mostly anaerobes. When we have our, just in brief, this cycle, when we have our normal metabolically active replicating bacterial cell, this vegetative cell, it comes across some sort of stress signal from the environment, and it turns on a bunch of genes that start sending it down this pathway of sporulation. Sporulation takes, depending on the species, a significant number of hours, 8 hours minimum, so this doesn't happen super rapidly. But what happens is you get this husk that forms around the genetic material, around the chromosome, bacterial chromosome. And then the rest of the cell starts dying away. So you can see some vegetative cells here, and then those cells, as they start to sporulate, those bacteria, you see that bright refractile spore that's starting to form. Where that spore forms is indicative of which genus it is. With Bacillus, when they form spores, those spores start forming in the center of the rod, which we'll see when we talk about Bacillus anthracis. But with Clostridium, like this is Clostridium tetani, those spores will form at the terminal of the rods, and it will cause a bulge or a swelling at that end. It looks almost like a tennis racket in some cases. And the rest of the cell can die away. When conditions become favorable again, those bacteria have ways of sensing it, even as spores, and they can germinate again back into vegetative cells. That process of germination takes much, much less time. That can happen in as little as 30 minutes. They can germinate back in vegetative cells. But once they do that, that vegetative cell, after it's come out of this spore form, it takes a while before it's able to replicate again. So there's a little bit of a lag phase that happens after germination. And just for clinical implication, as a reminder, these are difficult to get rid of in hospital settings, particularly with Clostridium. There are environmental implications for Bacillus that we'll talk about. They tend to force, when they're forming spores, they'll stay out in the environment for a long period of time. But when you come into contact with the spores and they germinate back into vegetative cells, that's when you're most susceptible to these pathogens, significant pathogens like anthrax and others. So the first one we'll talk about is Bacillus anthracis, which is like the mother of all biological threat agents. Select agent. One of the reasons why it's a select agent, in addition to the reasons that we've talked about with other select agents, which would be like the low ID50 and the ability to aerosolize it, is your ability to find it out in nature. So you can, Bacillus anthracis is a soil bacterium, and it's also what you'd call enzootic in livestock populations worldwide in herbivores. So enzootic is pretty much the same as saying something that's endemic in people. Demos meaning people, zootic meaning animals. So it's maintained in animal populations. When those animals die, the bacteria will sporulate, and those spores will go into the soil and contaminate the soil for decades, sometimes centuries, until another grazing ruminant will come along, and then as it's grazing, it will pick up those spores, and the spores will germinate and cause disease in those livestock animals. So that's the way it persists out in nature like that. When humans come into contact with anthrax, the route of entry determines what kind of form of anthrax you get. If you breathe in those spores, which can happen naturally or through biological warfare, you can get pulmonary anthrax. This picture on the bottom there shows people who would have a likely exposure to pulmonary anthrax when they're handling animal hides that have where the animal's been infected with anthrax, so they can breathe it in as they're handling those hides and get what used to be called wool sorter's disease, which you don't hear about much anymore. So that's one form. You can also, particularly if you're handling or near these animals who might have anthrax, you can get a subcutaneous inoculation if you have breaks in your skin and get cutaneous anthrax. And then lastly, if you were to eat meat from an animal who died of anthrax and that meat wasn't cooked all the way, you could get gastrointestinal anthrax. So we'll talk about all of those. In terms of prevention, we do have a vaccine for people. How many of you guys have the anthrax vaccine? Just about everybody. Okay. So these days, our vaccine that we get is it's a component or subunit vaccine. I think it's only has like the protective antigen component, Bacillus anthracis, in it. For livestock, we vaccinate livestock herds in the United States in an attempt to control natural transmission to people. And they get the, I think like either I think they get an attenuated vaccine of Bacillus anthracis, which would be problematic for us. So they get different types of the vaccine. Not everybody in the general population gets the anthrax vaccine though. We get it generally, as you guys probably all know, when we are at risk for, when we're deploying or going out somewhere where we're likely, or not likely, I shouldn't say likely, it's not likely at all, where we might be exposed to anthrax. Other people who would get anthrax vaccines would be veterinarians, people who work with livestock, maybe some lab workers. So it's not a vaccine that everybody gets. Okay, in terms of pathogenesis, this bacteria has two plasmids that are really, really important for pathogenesis, PX01 and PX02. You don't have to know those for the test, but I just want to explain why it's super important and why that makes this bacteria so virulent. When those spores germinate after you've acquired them through whatever route of transmission you acquire them through, when they germinate, those spores will fall away and the spores aren't encapsulated, but the actual bacteria need to express that capsule in order to survive in the human host. That capsule is encoded on that second plasmid, PX02, and if you knock that out, you have a strain that is entirely avirulent, does not cause disease. The other plasmid that is associated with virulence for Bacillus anthracis has a couple toxins that are super important. Edema toxin ET and lethal toxin LT, and it also has the protective antigen gene. So when it encodes and expresses all three of those plasmids or of those genes on PX01, you get these toxins that can do a lot of damage associated with Bacillus anthracis. The edema toxin causes dysregulation of cyclic AMP cells, so that contributes to edema. And then the lethal toxin disrupts cell signaling and causes ultimately those cells to die. When those two work in conjunction with each other, you get a lot of the pathology associated with Bacillus anthracis. And the protective antigen, which is not on this slide, but that's the part that when it's expressed, it forms a pore in target cells and allows these toxins to get into those cells. So that capsule is really important. It has a unique composition, this poly-D-glutamic acid composition. This is what Bacillus anthracis looks like when you grow it up. It's not difficult to grow. It grows pretty rapidly. This is a standard blood otter plate. It has kind of a distinctive edge to it. These are sometimes called Medusa head colonies, have like that frilly edge. But it's super fast and easy to grow. When I was with the AML, the First Area Medical Lab, which is an operational unit that uses microbiologists on their biological threat team, we would get a lot of samples of B. anthracis to work up, to be able to test and identify. And they would give us a strain that lacked the capsule, so it had the PXO2 plasmid removed from it, and we would work that up. But it grows basically the same. So this is an organism that I've grown up quite a bit, very easy to work with, which doesn't bode well for bioterrorism. And then when you do a gram stain on these things, they have a very distinctive morphology. Gram-positive rods, but as you can see here, you can see those spores forming right in the middle of the rod, the central location of the spores. So these bacteria are starting to sporulate. And they're also very large rods, so easy to see, distinctive boxcar morphology. These rods that are in singles or in long chains like this, sometimes described as like a necklace or string of pearls morphology. And we have electron micrograph that also is showing pretty nicely a bacterial cell that's in the process of either sporulating or germinating. It's hard to tell which way. For lab identification, in addition to culture and microscopy, we have PCRs that we can do on Bacillus anthracis, and we also have immunoassays that can look for things like the protective antigen or ET or LT toxins. All of those are possible. For that PCR, this is a target on a panel from BioFire that we would use for suspected biological weapons. So that panel will have something like 15 to 20 different targets on it. It's a multiplex PCR panel. Bacillus anthracis is one of those targets. Francisella tularemia is another. The Burkholderia are on there. So if there's a suspicion of that happening, this is a common target on that BioFire PCR panel. All right, talking about these presentations. The first form we'll talk about is the form where you are breathing in aerosolized spores. These spores will be breathed into your lungs, and then in your lungs, those alveolar macrophages in your lungs will take up or engulf those bacteria once they've germinated. The macrophages that have those bacteria engulfed in them will take them back to the lymph nodes in the mediastinum, so the mediastinal lymph nodes, and there they will replicate. So the lymph nodes are supposed to be an area where you do not have live bacteria, but these bacteria are not only live, but they're replicating in that area. This is around the time that you'd start getting some pretty severe symptoms. So this too, within the first 6 days, you might get some non-specific flu-like symptoms. And during that time, you have germination going on, which takes some time for bacteria to germinate, like I said. But it also, you have replication going on, and you have toxin production going on. By the time you have sufficient bacteremia in the lymph nodes, in the middle of that chest, so the mediastinal lymph nodes are sort of in between the lungs by the heart, an area that has a lot of vasculature and a lot of blood vessels. By the time you have all of that going on, some of that pathology is like respiratory failure, because the mediastinal lymph nodes are so swollen and distended at that point. That could be pressing on your heart. It can cause some chest discomfort from the extreme swelling that's going on and the edema from the edema toxin. So one of the classic ways to diagnose pulmonary anthrax is to do a chest x-ray and look for the widening of the mediastinum, like you can see on this chest x-ray. You haven't had radiology yet, but what you're looking at here is just that's the centric mediastinum on this chest x-ray. It is considered to be a hallmark for pulmonary anthrax. If these bacteria, so that's really severe, this respiratory failure, rapid onset sepsis, but if these bacteria get outside of the lungs and get into the bloodstream, patients can get septic and they can die of sepsis. Or those bacteria can go on to infect other parts of the body, like the meninges, and you get meningeal symptoms as well. So that's fairly common. I don't have the mortality rates on here, unfortunately. I'm not sure I know what they are for pulmonary anthrax. But we do have some historical data from some high-profile attacks that have happened with that involve pulmonary anthrax cases. I know a lot of you guys are pretty young, but does anybody remember the 2001 Amerithrax attacks? Okay, yeah, so with those attacks, we had, I think in around October, September of 2001, a terrorist who sent anthrax spores to a bunch of news stations, like 3 different news stations, ABC, CBS, NBC. And that was the first wave. I don't know if anyone got sick in that first wave, but then there was a second wave a month later where that same person or same entity sent more anthrax spores to some senators. So Ted Daschle was one of those senators and another one as well. That caused a couple people to be infected with pulmonary anthrax when they breathed in those spores. People that are opening the mail, these high-profile senators who open their own mail. So 17 people got pulmonary anthrax. 5 of those people died. And the people who died, again, these were like the mailroom workers. So it's really, really sad. And because of that, the intended target was to cause like wide-scale panic and confusion, and it disrupted mail service for a long time. So that was definitely a mess. And that was a deliberate use of anthrax spores to cause as a terrorist act. We also had a very well-known incident that probably occurred before any of us were born in 1979 in Sverdlovsk, Russia. Heard of that incident? It's referred to as the Sverdlovsk incident. But at that time in the USSR, in this city, which used to be called Sverdlovsk, now it's Ekaterinburg, they had a biological weapons facility. They had a faulty vent, or a vent that was removed during routine maintenance, and it allowed for all these anthrax spores to get released basically into a massive cloud. And then the breeze, which was blowing southeast, took this cloud of anthrax spores and wafted it down to a heavily populated area where around, estimates are hazy, but around 70 to 75 people died as a result of pulmonary anthrax from an accident. So, and that was like during the Soviet period, so they covered that up for years. Okay, so that was pulmonary anthrax. With gastrointestinal anthrax, different route of entry, very, very uncommon, but it is possible to have people get sick with gastrointestinal anthrax. This form is more common in livestock. So here we have livestock who are ingesting the spores that are in the soil. The spores will germinate, and depending on where that happens, you might get different parts of the GI tract that are affected. Ulcers can form in the mouth, the esophagus, or they can form later in the GI tract. Most of those spores will germinate once they hit the intestines after they clear the stomach. And when that happens, the ulcers that form in the intestines can oftentimes lead to sepsis because you have perforated intestinal barrier at that point. Very high mortality rates, nearly 100% in people if sepsis develops. This would be untreated gastrointestinal anthrax. In animals, this will kill animals as well. You can see this sheep and what a cow, I guess, some other ruminant. There's blood coming from the mouth, or they might have developed some ulcers in the mouth, and they would be bleeding probably in other parts of the GI tract as well. Let's see. The symptoms might differ according to where that happens, but that's basically the picture there. The last case of GI anthrax that I saw in ProMed was in Africa, I think in the DRC, where people had come across hippo, and this hippo had anthrax. They ate the hippo without cooking it because it was bushmeat, it was just available. But that, it happens. Like bushmeat in Africa, it's eaten a lot, and if it's not cooked all the way, you can get all sorts of exotic diseases. So that killed like 6 people who ate that hippo. But I'm not aware of any cases of GI anthrax in the US with people. Last form is cutaneous anthrax. This is the form that is by far and away most common in people. 95% of our anthrax cases are involve cutaneous anthrax. This is simply where you have some sort of scratch or laceration, usually on your hands, arms, sometimes neck, and spores, anthrax spores, enter into that open break in your skin, and then get into your bloodstream and germinate from there. Initially, that might present like a tiny little insect bite, a raised bump at the entry site. Eventually, that will develop and become a papule, and then a vesicle. The vesicle has fluid in it. That vesicle ruptures, and then that fluid dries out and becomes a blackened eschar. So an eschar is a black, dried scab, basically. It's associated heavily with anthrax, but anthrax is not the only disease that's associated with eschar formation. There are some other things that present that way as well. This is what an eschar looks like on a guy's neck, and you can see these photos after 3 days, 4 days, 15 days, 2 weeks after acquiring the infection, what that looks like. The eschar itself isn't painful, but the area around it, you can see some inflammation around that erythematous, reddening around that eschar. Mortality rates are much lower, maybe 20 to 25% if untreated. Really important to administer antibiotics early on. Exposure history and index of suspicion would be important in diagnosing cutaneous anthrax there. In terms of samples, this would be easy to find bacteria in any of these specimen types, but if you suspected it was anthrax, you would want to send that to a lab that's specialized and able to handle a select agent like Bacillus anthracis for workup. But any of these affected skin lesions would yield viable organism if it was handled correctly. And before you administered antibiotics, which might otherwise kill those bacteria. You can also pick it up by PCR. We have immunoassays that could look for the toxins for ET and LT and PA. So there are a number of ways to look for confirmed diagnosis of cutaneous anthrax. For the rest of the day, and then this is all I have for you today, I just want to go over this one case study of cutaneous anthrax in some detail. I think it's a nice time to do a deep dive because it pulls together, like this one case study that happened about 1.5 years ago, pulls together not just the clinical presentation, but how they diagnosed it. So a lot of lab stuff in here too. And it happened in Texas. So just for orientation, I'm not from Texas. I'm not super familiar with the geography here. But this is Austin in the Starrett Center, the capital of Texas. And then we've got a couple counties in between, and there's Bexar County right there next to that orange center. So here's Travis County, where Austin is, and here's Bexar County. And then there is an anthrax triangle in Texas, an area where anthrax is endemic here, that extends from Uvalde down just about to the Mexican border, Eagle Pass, and then up to whatever these counties are up here. So it's a fairly broad area, and this is where there are a lot of cattle out in that area. It's still very infrequent for people to get it, but cases do happen, and I don't know how much they enforce the need to vaccinate herds, but with this case in particular, we have a rancher that was somewhere living in and around this anthrax triangle in West Texas. And this rancher, the day before Christmas, on 24 December, had come across an animal, a lamb, that died suddenly on his ranch. So he handled it, he butchered it, and he proceeded to cook it and then serve it up. There were a bunch of other people involved who ate this meat, but he was the one who handled it. Okay. When he originally presented on the 4th of January, so this would have been like 10 days after exposure, potentially, 10 days post-exposure, he presented with fever, leukocytosis, a black eschar on his right wrist, and extensive edema and blistered lesions on his right arm. So out of that first sentence, what are some things that might tip you off to anthrax? The eschar, first and foremost. Yeah, he's in that area. Whoops. Well, there you go. He's in that part of Texas. His occupation is a rancher. That could be helpful. His age, maybe, maybe not. Yeah, go ahead. Yes, exactly. Never eat something where you don't know how it died is kind of a good golden general rule to follow, I think. His age, 50 to 59, I mean, that's getting a little bit up there, I don't know. Fever, leukocytosis. What is leukocytosis? White blood cells, so increased levels of white blood cells. Extensive edema and blister lesions. So he was febrile, elevated white count, and fortunately they did suspect anthrax. But the next slide, I think, goes into more detail. It didn't seem like that was their first suspicion. They initially suspected something else. What else? So the meat, when he did butcher that lamb, he cooked it well. So if there were any viable bacteria in that lamb, it was destroyed. You always have to worry about toxin, though. Toxin might be more heat resistant. But judging by the fact that the 5 other people who ate that, none of them got sick, there's probably nothing left to make those people sick. So GI anthrax, not a thing. He cooked it well enough. Good job. This is a lot of text, but again, like it's good stuff because it ties together so much. So hopefully you can see that on your screen. The font is a little bit small. But when he did originally go, he was seen by a GP, like a normal doc, and he was evaluated on New Year's Day. They thought he had a skin and soft tissue infection, so they prescribed this first-generation cephalosporin, cephalexin, and that was probably with the intention of clearing up an infection caused by staph or strep. That's probably what they were thinking when he presented initially. That's actually contraindicated for Bacillus anthracis. They have intrinsic resistance to beta-lactams, so that would be a bad thing to do. But they didn't know any better, so it's a reasonable thing. There's no judgment, but that would not be a good antibiotic to start somebody off with empirically for anthrax. Once his condition started worsening, that he wasn't responding to the antibiotic, he was moved to a hospital, and there at that hospital they took a more detailed clinical history. So at that point they were probably finding out things like his occupation, and maybe he was disclosing the fact that he had slaughtered this, or not slaughtered, he had butchered a lamb that had died of unknown causes, and other things that would have tipped them off. And then at that point, maybe his eschar started to develop into something a little bit more recognizable as containing anthrax. So they submitted wound swabs from the eschar. They took blood from him, initiated antimicrobial monotherapy for non-systemic cutaneous anthrax. So his symptoms, he was probably febrile at that point if he had systemic symptoms. And they moved to dual therapy, cipro and clindamycin. I don't know how well that worked. But one of the points is that he was already on antibiotics by the time they took wound swabs and blood, so that might have impacted the ability to recover viable bacteria from those specimens. His condition worsened, he got transferred to another hospital, so the systemic symptoms are still progressing with severe edema and malaise. This was handled by, at least the specimens were transferred to the Texas Department of State Health Services lab. So that's a lab that would be equipped to handle something like Bacillus anthracis. They did a PCR test and they found DNA on those wound swabs. So it was positive there, but nothing grew out from culture. No viable bacteria. Meanwhile, they were doing an investigation with the animals. So the lamb, they took some specimens from those lambs, and a couple days later, on the 6th and the 11th of January, 2 ewes, those are female sheep, died on farm with ocular and nasal hemorrhage. So, pretty good indication that those animals had anthrax. And nasal swabs collected from those animals after 12 hours, or 12 hours after death. Why is that important? 12 hours after death, do you think? Yeah, as soon as an organism dies, the chemistry of the body changes significantly and a lot of bacteria die. So if you're collecting a specimen post-mortem from anything, animal or human, 12 hours would be a time point where you might not be able to recover any viable bacteria. So they collected those specimens pretty late, sent it off to another lab, state lab, to culture, weren't able to culture anything from those. But finally, with these other assays, that's when they got their definitive diagnosis. So they sent the patient's serum off to the CDC They did paired sera, which means they sent the sera off for, like, is collected early on in the infection during the acute phase. So initially you get IgM, right? Usually around like 7 to 10 days, 7 to 14 days, IgM. And then you do class switching and you get some IgG starting between like 10 to 21 days, perhaps. So you do paired sera to show that there's an increase in the titers of IgG. And that was one of the things they used to help diagnose them. They also did quantitative ELISAs on the toxins as well as the protective antigen. They quantified that. This is kind of interesting. They had lethal factor concentration measured at almost 12 nanograms per mil in the acute serum, one of the highest lethal factor levels ever measured in a patient with cutaneous anthrax. I don't know how many specimens they get, probably not that many, but this guy had a lot of this toxin in that specimen. The cooked meat, when they did PCR on that, did they find, was it PCR positive? Yeah, it was PCR positive on the cooked meat, so they were able to find DNA in that cooked meat, but nothing grew, of course. And then this, very briefly, we have, again, a comment about the use of cephalosporins in Bacillus anthracis. We didn't know any better, but they do have that beta-lactam, intrinsic resistance to beta-lactams. They produce a chromosomally encoded beta-lactamase, so that's why that was not working. Another point to underscore is that there's no safe way to prepare meat for consumption from an animal that has died of anthrax, especially if you undercook it. But if you cook it well enough, as in this case, like nobody got sick from the 5 people who ate that meat. And then lastly, in that anthrax triangle, this occurred on a farm next to where the last case happened. 2019, there was another cutaneous anthrax case that happened. 25 culture positive animal cases in that area. That's a huge number of animals that were positive for the anthracis. And I don't know what they did with those animals, if they culled them or something else. Avoid processing carcasses of animals that died unexpectedly, and then also this point about vaccination here, there was no clear history of routine vaccination against anthrax for this bird. So, yeah. His labs are all right here. I know that's really small print. This is just an FYI if you want to take a look at the timeline, which is really, really interesting from the time that the lamb was butchered all the way up to some of the last results that came in from mass spec on these toxins. But some interesting things to note here, you haven't had some of this information you'll get next semester in ClinLab on how to interpret lab results. But one thing that's evident is the neutrophils when we're talking about leukocytosis. These neutrophils were very, very high. The reference range is indicated here. So neutrophil percent, 94%, where the reference range is 42 to 75%. So even without having gone through ClinLab, you can kind of get an appreciation for how severe this infection was and some of his lab results. Okay, so I know that's a bit of a deep dive, but again, hopefully that ties together some disparate points in how they went about this investigation for anthrax and how that just happened basically like one county to the west of us in the anthrax triangle. Any questions about anthrax? Okay. One more, this is the last slide, Bacillus cereus, also in the same genus, also spore forming. This is definitely another toxin producing bacterial species. It's associated heavily with food poisoning. There are 2 forms to be familiar with, emetic form and diarrheal form. In terms of how this bacteria gets into food supplies, it's just a soil environmental bacteria, and it's very difficult to eradicate it from our food chain. So it gets into some of these food items probably early on in the process, and if that bacteria is viable, it can start replicating, and you can get this classic diarrheal form that's caused by the bacteria itself. So, living bacteria, the incubation period, this is very similar to some other diarrheal infections that we talked about earlier, like staphylococcal enterotoxin B versus, which is like a toxin-mediated form versus an actual infection. But the incubation period is longer because you have an organism behind it. And then in terms of symptomology, diarrhea is present in this presentation. This also lasts about the same length of time as the emetic form. And if there are toxins produced by the strains of Bacillus cereus, they are heat labile, so they're destroyed by heating processes. With the emetic form, you do not have to have any bacteria that survive in that food in order for this to cause disease because it's toxin mediated. So this bacteria produces those toxins, and it's implicated a lot with fried rice. That's like the most common association with this emetic form. But those toxins in the emetic form stimulate vomiting, very rapid onset, to be within 30 minutes, anywhere from 30 minutes to 6 hours. And again, those are going to be heat and acid stable. So if you have a large batch of food, whether it's fried rice or something else that it's contaminated with that toxin, you can reheat it gently. Maybe it's not to the point where it's like steaming and super hot, but a gentle, mild reheating, and the toxins will retain their activity and be able to cause that presentation. If you really nuke it long enough, you will destroy the toxin and you will not get disease. But the cases we get that are associated with the emetic form of Bacillus cereus are associated with not getting it hot enough, not getting the food that's contaminated hot enough in order to destroy the toxin. Those are the foodborne presentations. Also, there are a couple other infections implicated with Bacillus cereus. Because it's a soil bacterium, sometimes you can get this rubbed into your eyes, and you can get keratitis, which is an infection of the outer surface of the eye, or you can get endophthalmitis, which involves like the aqueous humor and vitreous fluids in the eye. It's a deeper eye infection. And also occasionally IV catheter-related sepsis is possible. But primarily you want to focus on these food poisoning forms that are fairly common. That's it for today. Any questions? Okay, we'll pick up tomorrow talking about Clostridium. I'll see you guys tomorrow at 10. Thank you.